So we'll be continuing our program here today. This next talk is going to be our young investigators or emerging investigators. This is something that I think is probably one of the most important talks we'll hear today. We really agreed as a board yesterday in our board meeting that one of the most important things we can do at ALD Connect is make sure that we are continuing to encourage and fund young investigators to make sure we are harnessing that next generation of genius minds, like all the minds we see here, like Hemmo right here. So very, very pleased to announce that we funded our first round of emerging investigator grants in 2022. It's a very important program, very important initiative of ALD Connect. And the goal of the program is to, as I said, introduce graduate students, medical students, postdoctorate fellows to our field and foster their continued learning and interest in ALD. So we awarded 3 grants in 2022 with the condition that each awardee has to come here and present, tell us what the hell they're actually doing. So we are very excited to hear what each of you have found here, and we are thrilled to welcome our first speaker, who will be Chenxu Li. Next speaker after that will be Hemmo Yska and April Rickle to hear the updates of their projects. So first, we have Chenxu Li.
Hi, everyone. I'm very pleased to see you all here today and share my research finding in the field of adrenoleukodystrophy. My name is Chenxu Li. I'm a PhD candidate from University of Minnesota, Center for Orphan Drug Research. And before my presentation, I would like to express my sincere gratitude to ALD Connect for providing this Emerging Investigator Award and holding this great event, as well as providing me the opportunity to present my work here today. So first of all, I would like to quickly go through some very basic information that most of us are already quite familiar. So ALD is caused by mutation in the ABCD1 gene, which is a gene encoded peroxisomal ATP binding cassette transporter protein. And the deficiency in such transporter protein results in metabolic disorder of peroxisome and lead to the storage and accumulation of very long-chain fatty acid, predominantly hexacosonic acid, in plasma, brain white matter, spinal cord, and adrenal cortex. So clinical manifestation exhibit substantial variation depending on the severity of the disease as well as the age that being affected, and the 3 main common phenotypes of ALD, including Addison's disease, adrenomyeloneuropathy, and cerebral ALD. And it is important to note that currently there is no established genotype-phenotype correlation, as patients with the same gene mutation can develop distinct phenotypes. Well, oxidative stress is defined as a pathological condition that caused by redox disequilibrium and leading to an excess of reactive oxygen species, which cannot be adequately neutralized by the cellular antioxidant defense system. Mitochondrial dysfunction and oxidative stress are tightly dependent on each other. Mitochondrial damage is not only one of the main contributors of oxidative stress, but also one of the main consequences of oxidative stress based on the high dependence of mitochondrial function on the redox-sensitive targets, such like intact membranes. So increased production of reactive oxygen species, endoplasmic reticulum stress, and at least in part, the transcriptional dysregulation as well as abnormal RNA processing are all consequences of mitochondrial dysfunction and oxidative stress, and ultimately contribute to the death of motor neurons. Here on the right-hand side, in ALD condition, mutation on ABCD1 gene results in elevation of very long-chain fatty acid, and those very long-chain fatty acid will then triggers oxidative stress and production of reactive oxygen species within cells and within mitochondria. And this makes the big consequence of mitochondrial dysfunction reflected by loss of ATP production, inhibition of mitochondrial biogenesis, as well as axonal degeneration. So currently, the approved therapy for ALD, including hematopoietic stem cell transplantation as well as gene therapy, are only designed and intended for treating cerebral ALD and need to be performed in the earlier stage of disease. However, there is no approved therapy that can be used to delay or arrest the disease progression in all individuals with such gene mutation. Literature evidence indicate that monounsaturated fatty acid can decrease viral long-chain fatty acid content in cultured skin fibroblasts derived from ALD patients, presenting the opportunity to intervene disease progression using dietary fatty acid. Well, as one of such dietary fatty acid, mavonic acid is a monounsaturated fatty acid with 24 couple chain and single double bind. It is naturally occurred and synthesized endogenously in our body as a main component in the white matter of brain as well as myelin sheaths. Mavonic acid is closely associated with the development and maintenance of brain, as well as biosynthesis and improvement of nerve cells. And also, mavonic acid has been found to activate antioxidant defense system, thus can act as a neuroprotective agent in the brain. And most importantly, we've seen the importance of mavonic acid in the underlying mechanism, highly based on the observation of decreased mavonic acid level in postmortem ALD brain. So with those information, we hypothesize that mavonic acid exhibit therapeutic efficacy in targeting viral long-chain fatty acid-related pathology, protecting from cellular oxidative stress, and improving mitochondrial function via various mechanisms of action. And also, we propose that mavonic acid is able to improve mitochondrial function through both direct and indirect effect by reducing the onset of oxidative stress. In order to test our hypothesis, a series of well-designed in vitro experiments have been conducted in our lab to test the therapeutic effect on each specific target, as I mentioned above. So first of all, in our published paper, we look at the biochemical effect of mavonic acid in ALD cells. So here in both figure A and figure B, the different shape of the dots refers to different cell line and different color refers to different fatty acid. And both figures shows the changes of different viral long-chain fatty acid following the treatment of either erucic acid, which is in figure A on the left-hand side, or following treatment of increasing concentration of mavonic acid, which is shown in figure B on the right-hand side. And here, I would like to bring your attention on these orange lines, especially on the right-hand side, which refers to the changes of C26, which is a biomarker of ALD. And our study indicate that mavonic acid is able to significantly and biochemically reverse the accumulation of C26 in ALD fibroblasts, whereas have almost no changes, no impacts on normal health fibroblasts. And furthermore, we examined the ATP content in different ALD cell lines, and by treating the cells with either erucic acid or a different concentration of mavonic acid, we find that in a general perspective, mavonic acid is able to increase the ATP content in ALD fibroblasts compared with the control group. And then our purpose is to investigate the effect of mavonic acid on cellular oxidative stress. To test this hypothesis, different fluorescent reagents were used to stain the cells and enable different research purposes. So first of all, in this figure, the green signal refers to a fluorescent dye called CellROX, which enable the detection of total reactive oxygen species in live cells, whereas the blue signal is DAPI that we use to localize and quantify the nuclei. So here, the quantitative results was shown on the left-hand side, and we find that mavonic acid is able to significantly reduce total reactive oxygen species in ALD fibroblasts, thus serving as a potential protector from cellular oxidative stress. And further, with similar experimental design, we use another compound, fluorescent compound called mitoSOX to enable the detection and quantification of mitochondrial superoxide. And here we find a similar results as shown in this figure, as well as the box plot, that neuronic acid can also specifically targeting and reducing mitochondria-derived superoxide in ALD fibroblasts. And then because we have previously seen that treating the cells with neuronic acid is able to increase the ATP content, we would like to further decipher this perspective to understand mitochondrial respiration with or without neuronic acid treatment. So first of all, we would like to confirm the pathological disease condition of ALD cells. Mitochondrial stress test was completed by my colleague, Dr. Marcia Turluk. Seahorse assay was conducted in live cells using XFE extracellular flux analyzer to enable a real-time assessment of cell respiration parameters in either health-controlled fibroblasts, which is shown in blue box, versus in ALD fibroblasts, which is shown in dark red box. And here, in each specific panel, it indicates different respiration parameters. And our results find that mostly of the parameters will show a significant decrease in ALD fibroblasts compared with health control cells, indicating a disease condition of mitochondrial function. And further, we would like to test the therapeutic effect of neuronic acid in those ALD fibroblasts. So similarly, each panel refers to each respiration parameter, whereas within each specific panel, from a left to right sequence, there refers to control group, vehicle control group, and different concentration of neuronic acid treatment group. And our study indicates that treating the cells with neuronic acid results in a significantly improved basal respiration, ATP production, maximal respiration, and spa respiratory capacity in fibroblasts derived from ALD patients. And subsequently, we are interested in whether those effect comes from the effect of neuronic acid on any specific mitochondrial metabolic pathway. So it is important to note that the TCA cycle plays an important role in our body, engage in the catabolism of biomolecules as well as energy production. And carbons derived from either glucose, as I show in this figure, carbons derived from either glucose, fatty acid, or glutamine will then enter the TCA cycle, and which in turn generate ATP to support each specific activity within our body. And by using specific inhibitors with either UK5099, atomoxir, or betas, we are able to inhibit each of the specific pathway and evaluate the effect of neuronic acid in rescuing those pathways. And the result is shown in this figure. So the upper figure indicates the basal respiration, whereas the lower figure indicates the maximal respiration. So first of all, we find that after treating the cells with neuronic acid, we find a significant increase in oxygen consumption rate for all three pathways for both maximal and basal respiration. And then by applying inhibitors to inhibit specific pathway and compare treating with neuronic acid to evaluate the rescue effect of neuronic acid on those pathways, there is a general increasing trend of neuronic acid in each specific pathway. So all of this information come up with a conclusion that neuronic acid is potentially beneficial to all mitochondrial metabolic pathway without only acting on one specific pathway. So as a conclusion of our research, our study demonstrates that neuronic acid can be a promising therapeutic candidate in targeting different mechanisms of action, but to be benefit to ALD patients. And furthermore, the effect of neuronic acid on mitochondrial function, oxidative stress, as well as immune regulation can be potentially beneficial to patients with not only ALD but also other neurological diseases. And last but not least, I would like to express my appreciation to my colleague, Dr. Marcia Turluk, and my advisor, Dr. Rena Carthar, for all the support and encouragement in my research project. And I appreciate Dr. Li's lab, Dr. Donta lab, as well as Center for Orphan Drug Research from University of Minnesota to provide the instrument as well as necessary technical support to complete my research. And I greatly appreciate all the following organizations, especially ALD Connect, to provide the financial support to complete our study. And I appreciate for your patience to listen to my presentation. And now I would like to take any questions. Thank you.
Thank you, Chenxu. Open it up to questions. Come on, these are emerging investigators. There we go, Bella.
Hi, thank you very much. Excellent talk. My name is Bella Turk from Kennedy Krieger Institute. We previously did some work looking at antioxidant capacity, so I love the work that you're doing. I have a specific question. Did you look at any specific enzymatic antioxidant capacity? It would be really interesting if there was a difference between BPTS and edemoxir.
Thanks for the question. We haven't looked through this perspective yet, but I think this is a very good point for our future step. Thank you.
Yeah, I want to compliment you on your presentation, particularly since you don't even look old enough to drive. But on your graphs of the mitochondrial oxidation, the first one, there were several graphs, and go back one more. Yeah, okay, there. Mm-hmm. The one on the second row to the right, where it's kind of reversed, the ALD. Okay, graph number F. The ALD cells have more oxidation. Can you explain what that anomaly is caused from?
Oh yes, even the figure F, right? This is the changes. Oh, this is the level of non-mitochondrial respiration, and because it's like the respiration comes from non-mitochondria, we thought it is possible that as a compensating effect because the mitochondria has been dysfunctioned, and we just come up with a hypothesis, and we haven't test this specific aspect in details, but I think this is a very good point to catch up with. However, we do find that most of the important parameters, including ATP production, basal respiration, and maximal respiration, are all significantly decreased in ALD condition. Thanks.
Yeah, congratulations. Really, really nice talk and really nice work. I have a question that's maybe predictable because this really makes me think of Lorenzol, because C24-1 is just 2 carbons longer than uric acid and C18-1 that are the ingredients of Lorenzol oil. Yeah. So what would be your next steps now from here? So is this going to give us an answer about Lorenzol oil, which is we still have no idea if it does anything or if what it does?
Oh, yes, yeah, for sure. So here in this presentation, I only show the results from my in vitro work, but in parallel, we also conduct a lot of in vivo study using ALD mouse model derived from Kennedy Krieger Institute and provided by Dr. Trailan's lab. And we, by assessing the effect of neuronic acid in a short-term period, we do find a significant reduce of C26 LPC, which is the diagnostic marker of ALD, as well as a significantly reduced C26 in plasma, in both plasma and in tissues. And we thought this is a good indicator of that neuronic acid is a promising candidate. And meanwhile, because I also have a group of mice treated with uric acid, which is the active component of Lorenzol oil, and we do find some cardiotoxicology in uric acid treatment group, but not in neuronic acid treatment group. And because another another concern, another perspective of we trying to test neuronic acid is because it is actually in combination with other compounds as a supplement to improve nerve cells and memories. It's already cells in market, so we thought this might brings a perspective that neuronic acid might have less safety concern than uracil acid. And the next step of our research is to develop long-term assessment of both safety and efficacy of neuronic acid in animal model. And if everything goes well, we will go further for clinical trial.
Oh, that's really
Yes.
promising. When you say tissues, do you also mean the brain of the mice?
Yes, this is unpublished results, but
You can share. We
yeah,
won't tell anybody.
Sorry, but we do analyze different tissue compartments, including brain, heart, kidney, adrenal gland. Yeah.
Okay,
Thanks.
great.
Any other questions? All right, let's give a round of applause. Thank you so much, Chenxu. And I did want to make a quick note. This year we did something different on our agendas. I know we didn't print them this year, but they are available online. We do have a layman's explanation for each of these talks. So, you know, if you're a layman and you want to understand a bit more of what exactly these genius minds are talking about, these explanations can really help with that. Okay, so our next emerging investigator hails all the way from the Netherlands. He will be talking about attitudes of patients with ALD towards sex-specific newborn screening. Let's give a warm welcome to Hemmo Yska.
Yes, thank you everyone, and thank you, Greg, for introducing me as a genius. That's very kind of you. So this is going to be a bit of a different presentation than the previous one, because it's a lot more clinical. My name is Hemmo Yska. I'm a medical doctor from the AMC, and I'm a PhD candidate in the Department of Pediatric Neurology with Mark Engelen. And my talk focuses on the attitudes of affected individuals towards newborn screening for ALD. So I think this is going to be known information for everyone, but just very quickly. ALD can manifest in several ways. And in boys and men, we can see that patients develop cerebral ALD, which is a leukodystrophy, which you can see here with a rim of contrast enhancement. And if you're early enough, you can treat this with stem cell transplantation. That can really be life-saving. The second manifestation is adrenal insufficiency, which can also be treated by steroid replacement therapy. And then both men and women can develop spinal cord disease for which there currently is no treatment, and it can only develop at an older age. So if you can find ALD at a very early stage, it can really be potentially life-saving, as we've also heard from the patient talks a few hours ago. Because of the stem cell transplantation, because of the steroid replacement therapy, which is why in 2013, the state of New York was the first to initiate a screening program for ALD, which led these two gentlemen, Stefan Kemp and Mark Engelen, to initiate these steps as well in the Netherlands, which led in 2015 already to the advice from the Dutch Ministry of Health to also add ALD to the Dutch newborn screening. With one side note, which was that the Dutch Health Council advised to only screen boys. And this principle is referred to as sex-specific screening. Now, the reason why the Dutch Health Council advised this is this set of principles, which are called the Wilson and Jungner criteria. And these are pretty old criteria, but they are still internationally accepted. And they're kind of the rules by which a disease should abide by in order to be eligible for newborn screening. And the problem with girls with ALD is the second point, highlighted in red, which is that there should be an accepted treatment for patients with this disease. And boys and men you can treat with the steroid replacement therapy and with the stem cell transplantation, but this option is not possible for women who only develop the spinal cord disease. So that's why the Dutch government decided not to screen girls. I think there are some pretty good arguments for this. For example, if you're a girl and you get the diagnosis at a very early age, it kind of makes you a patient in waiting because you don't have any symptoms and probably you're going to be 40 before you develop the first symptoms. You also have a right not to know as a patient that you have a disease. It can have a negative impact in a financial way, for instance, difficulties in getting life insurance. So there's some pretty good arguments, in my opinion, on why not to screen girls. On the other hand, there's also a set of arguments from a more ALD experiential perspective why you should know at an early age if you're a girl that you have ALD. For example, you can make decisions on your future. It decreases the time before you get the diagnosis if you have complaints, so the diagnostic odyssey. Maybe you can take some steps on family planning, extended family screening. So there are some pretty good reasons to plead the other way as well. So during the introduction of ALD to the Dutch newborn screening, this study investigated by Van der Pol et al. Investigated the attitudes of a general population of parents of newborn children towards the addition of ALD to newborn screening. And the conclusion was that in general, everybody was very positive about it. But strikingly, 86% of this population thought that both boys and girls should be screened for ALD. So we know something about the general attitude, but what we don't really know yet is how do affected individuals themselves feel about, how do patients feel about this? And this is a bit strange because I think ALD patients are maybe the most important stakeholder group. So that's why we decided to investigate the attitudes of patients with ALD towards sex-specific screening. And we did that by asking patients in the Dutch ALD cohort, which is about 120 patients, how they felt about that. We did that by means of a questionnaire. And basically what we asked is who should be screened for ALD? Should it be only boys? Should it be both boys and girls? Or should nobody be screened, actually? We also investigated the influence of background variables, and we investigated the motives behind this decision. And we did that in two different ways. First of all, by means of open text responses. So just, what is your main reason why you have this screening preference? And the second one was by providing 19 statements, and then people could indicate their level of agreement to this statement. So what do these statements look like? Well, they were on a 5-point scale, with 1 meaning no agreement at all, and 5 meaning full agreement. We split the statement into 4 categories. The first one being arguments in favor of screening newborn boys, which I will get back to examples of these statements later. The second one, arguments in favor of screening newborn girls, arguments opposed to screening newborn girls, and arguments opposed to screening newborns in general. So on to the results. We had a response rate of 64%, which is quite high for a survey study like this. We had 38 men and 29 women participate, and we found that 80% prefer to screen both boys and girls for ALD, 20% prefer to screen only boys, and there was nobody that didn't want to screen anybody for ALD. Onto the motives. First of all, the open text responses. Within the group of people that felt that only boys should be screened, about 50% felt that the main reason for that is because boys develop more severe symptoms and that girls develop less severe or no symptoms at all. If we compare that to the group who prefer to screen both boys and girls, we see that about 1/3 of all people feel that it helps provide an early diagnosis. 1/4 felt that it facilitates treatment and monitoring, and about 17% felt that it helps with family planning. And I think what is important to realize with these statements is that actually quite often people didn't really indicate to what sex this referred to. So for instance, if someone says it helps to provide an early diagnosis, do they mean for women? Do they mean for men? For both? We don't really know that. So there are some problems with these motives. And in general, I think I found a really striking and exemplary statement from one of the people that participated is that the consequences of a girl not knowing you have ALD can be very severe. And with newborn screening and its results, you can get better support and disease monitoring. And this is something that we heard a lot actually in these open text responses. So onto the other way that we investigated the motives. These are the statements. There are 4 categories, like I said. And this is the category of arguments in favor of screening newborn boys. And unsurprisingly, everybody really agreed to this. So the score of 5 on average is the highest, and 1 is no agreement at all. And here you can see that for all statements, both people in favor of only screening boys and people in favor of screening both boys and girls, they really agree to this. And this is not surprising because nobody is not in favor of screening newborns at all for ALD. So that's unsurprising. The second category of arguments opposed to screening newborns in general, also not surprising, nobody agrees with this. The statement ALD should not be detected early within the Hilbrick because it's too expensive for society, people agreed the least to that statement. Where the groups kind of start to diverge a little bit is in the statements related to screening girls. So within this category of arguments opposed to screening newborn girls, you see that the group who is in favor of screening boys and girls does not agree to these statements. So for example, the statement ALD should not be detected early in girls because they almost never develop life-threatening symptoms. Nobody really agrees with this. And the group in favor of only screening boys, they have slightly positive attitudes towards these statements. What I think is the most interesting category of statements is this one, which is the arguments in favor of screening newborn girls. Unsurprisingly, the screened. But if you take a look at the mean responses of the respondents that feel that only boys should be screened, for some of these statements, they do also slightly agree to this. For example, the second statement, which is ALD should be detected early in girls so that parents can make better decisions on family planning. These people feel that only boys should be screened, but they do kind of see the advantages of this. So I think this really illustrates the very difficult balance on making a decision, because in the end, they do feel that only boys should be screened, but they do see the advantages of screening girls as well. I can be really short about the influence of background factors because we did not find a significant difference or influence of sex, age, the death of a family member, an alternative lifestyle, and the experience burden of disease. So we did, within our cohort, no clear suggestion on any background influence. Which leads to these conclusions. Patients with ALD have a positive attitude towards the addition of ALD to newborn screening. The majority has a preference for screening both boys and girls because it is important to know before the onset of symptoms, and it is important for family planning. A minority has a preference for only screening boys because boys develop more severe symptoms than girls, and there is no treatment for girls at the moment. And we did not find an influence of background within our cohort. So to put these results a little bit into the context, because the Netherlands is not the only country, as we all know, who is involved in newborn screening for ALD. The United States, obviously. I think this graph, I'm pretty sure that this graph is not up to date because I see a few gray states which have started already. But also Taiwan screen for ALD. And interestingly, there's, to my knowledge, not really a big debate on whether to only screen boys or both boys and girls. And you could say that this is a little bit in conflict with the Wilson and Jungner criteria, which once again, these are not just European criteria, they're really internationally accepted. But I think the thing is that you have two interpretations of the word treatment in these criteria, where if you take it very narrowly, you say, well, we're screening the newborn, and the newborn cannot be, the girl will not die from this disease, so you can also not save her. Whereas if you take a more broad interpretation, maybe symptomatic treatment, maybe family screening, or giving the opportunity to make reproductive choices can also be seen as treatment in a way. And there's a few countries that are currently also in the process of the addition of ALD to their newborn screening. And out of these, I know that Japan also tends to go in the direction of screening only boys. And I think the other countries and the other states in the US, they're more in the direction of screening both boys and girls. So are these results very surprising? In my opinion, no. Because if you ask people with a disease, do you think that people should be screened for this disease? I think the majority will say yes. So in that sense, it's not really a surprise, and it's not a definitive advice on who should be screened. But what I think it is it's an important study because ALD patients are such important stakeholders. We found that a similar proportion of patients is in favor of sex-specific screening as a general population. So that's also important to realize. We got insights into the motives of these people. And what I found pretty surprising is that we didn't find any influence of background. Because previously or prior to this study, we hypothesized that maybe women would be more inclined to screen girls as well, because they can relate their experiences more to the disease, and they want to give the chance to girls to also do that. But we did not find this influence. So maybe it's just in the cohort, but could also just be a real result. So how generalizable are these results? Well, I think what's good is we have a relatively large cohort for this disease, and we had a high participation rate. What is maybe a bit unclear is how comparable these results are to other countries. The survey is freely available, so if anybody feels that they want to perform the study as well in another country, I highly recommend you to initiate a similar study, because I think it's too important not to do. And with that, I would like to thank the project team. This is Liedewij Henneman, who's a professor of reproductive medicine. She really helped a lot with the survey. Rinse Barends, who initiated this project when he was still a PhD student. Obviously, Mark and Stefan. And last but definitely not least, thanks a lot to ALD Connect for giving this emerging investigator grant. It really helped with setting up this project, and I really appreciate the confidence in this work. And I'm also happy to say that the study has been recently published in the International Journal of Neonatal Screening. So it's freely accessible to everyone. And thank you all for listening, and I'd be happy to answer any questions.
Thank you, Hemmo. All right, questions?
Thank you. That was very good, and I understood everything you said, which was great.
I tried my best.
One question. So when you were talking about the demographics, that's a woman answering. Did you look at whether or not it's a woman that was diagnosed versus if it was a man diagnosed?
Yeah, you mean if it was a woman who was primarily diagnosed because of symptoms, or whether it was like extended family screening?
Yeah, I wasn't sure on the demographics if you were splitting it up, saying all the women, regardless of whether or not their family member that was the first diagnosed was a man or a woman, answered this way.
Yeah.
Or if it was separated out, like a dad of a girl might think differently.
Yeah, no, we didn't do a statistical analysis with that, but we did do a lot of descriptive statistics comparisons, and for almost all background variables, the distribution between the screening preferences was very similar. Yeah.
Any other questions? Amber.
It's interesting the way you describe. First of all, it's great work. Thank you for doing it. What is quote unquote a treatment? So we talk about that if a woman knew in advance, then she could exercise more reproductive freedom. But there wasn't anything in there, the consequence medically of having a child with ALD. How many have committed suicide? How many marriages have been destroyed? Depression? Like, if you could prevent that, I would conjecture to say that's a treatment.
Yeah, I think that's a very good point. I think it's for the public health decision perspective, I think that's a very broad interpretation, but it is very important. Yeah, I definitely agree. Yeah.
There we go. Oh, okay.
Perfect.
So, you know, in terms of this question of treatment, even if you're being like very strict about that definition, my hope certainly would be within the next few years, we have treatments available for women. So kind of projecting forward, what would that look like, or what would be the timeline for the Dutch public health ministry to potentially adjust newborn screening to include women? Like, has that been a conversation at all? Do you know?
Yeah, I think there would really have to be already a treatment in place for women. But for instance, with the gene therapy that we're working on now with together with Swan Bio, if that will be a really effective treatment, I think probably the Dutch Institute of Health will be a little less critical on the definition of treatment. I think the thing is, because we keep identifying more and more rare diseases, and more is possible as well. You have to draw the line somewhere on the definition of treatment. And I think this is quite a strict line, so to say. I know that another thing that the Dutch Institutes of Health were kind of afraid of is that currently the participation rate is very high in the newborn screening in the Netherlands. I think it's almost 100%, like 99, 97% or something. And that if you would decrease the rationale of the newborn screening by having a broader interpretation of the word treatment, maybe it gets more difficult for people. Maybe they don't really understand anymore. And then they will say, we don't want to participate anymore. I know that's a big reason as well why they're being pretty strict about it. Yeah.
All right. Round of applause for Hemmo. Thank you
Thanks,
so
Rickle.
much. Our next speaker will be April Rickle. She'll be discussing testing the impact of the drug liraglutide in a fruit fly model of ALD. Hopefully I pronounced that correctly.
Hi, everybody. Thanks for coming to my talk. I just wanted to start out by thanking ALD Connect for both funding this research as well as giving me the opportunity to speak with all of you today. I'm going to be talking about testing liraglutide treatment in a fruit fly model of ALD. So first off, why do we care about fruit flies? There's a couple different reasons that fruit flies are very useful for research. First off, Drosophila fruit flies do have a homologue for ABCD1. It is also called ABCD1, and it is equivalent to human ABCD1. They also lack ABCD2. So if you knock out ABCD1, they don't have any means of compensating for that loss of ABCD1. This is a clean knockout system. They also have a short lifespan, so we're able to see development of the ALD phenotype in our flies in 5 weeks. And that allows us to work very quickly. And you also have very easily accessible brain tissue. You can dissect out the brains pretty quickly once you have practice, and you can stain them for whatever you want. You can blend them up and use them for metabolomics or transcriptomics. There's a lot of options there. There are also established phenotyping assays that are accepted within the field for detecting and quantifying neurodegeneration. So it's not unusual to be studying a neurodegenerative disease in fruit flies. And there are a number of different ABCD1 mutants that are available already through stock centers. So you can test multiple different genetic backgrounds. So to begin, the 2 mutants that I'm working with in my research were both generated by other labs, and they were generated through insertion of a transgene into the ABCD1 gene. So one mutant was inserted before the beginning of the translated region, and one was inserted into the middle of an intron in the protein. And despite these 2 mutants being in different genetic backgrounds and having different insertion points, we can see through qPCR that the expression of ABCD1 is knocked out in our mutants compared to wild type. So let's look at the phenotype of these mutants. First off, just looking at lifespan, ABCD1 mutants do show a reduced lifespan compared to wild type. You can see on the Kaplan-Meier curve here that ABCD1 mutant flies begin to die off much more quickly than wild type flies. And while wild type flies have a median total lifespan of about 60 days, ABCD1 mutant flies have a median lifespan between 40 and 50 days. So what else can we say about what their phenotype is? Well, we can look at if it would let me There we go. We can look at a climbing assay as well in order to quantify their level of neurodegeneration by measuring how well they're moving. So here's data from that. You can see that at 1 week of age, the flies, the mutant flies and wild type flies don't really have any significant difference in ability to climb from one another. But by 5 weeks of age, you have a significant difference in climbing ability between our wild type and mutant. Notably, it drops quite a bit. It drops quite a bit towards the middle of their lifespan. This is not their full lifespan that we're looking at, but this is when we start to see the our mutant flies stop moving so much, so we don't usually measure beyond this point. So this climbing data shows us that that our flies are experiencing neurodegeneration, but we can also look more directly at neurodegeneration by dissecting out the fly brain and staining it. So this is a representative image of a fly brain. It's from a 5-week-old mutant fly, and it's stained for F-actin, which is a structural component of cells. This should make every cell in the brain light up. Using this, we can look for voids in the staining where there has been a loss of cells, which is indicative of neurodegeneration. So here we see 3 different lesions in the fly brain where there has been a loss of cells. And to note, it is different from these physiological holes in the fly brain, which are more akin to ventricles in human brains. And using this staining, we can quantify neurodegenerative lesions in our fruit flies. So we can see that our mutant 2 flies have a significantly higher number of lesions per brain compared to wild type. And the lesions that the wild type flies do develop with age are smaller, both in terms of volume and surface area compared to the mutant flies. Using our ability to dissect out brains, we also performed whole brain metabolomics. So we did an untargeted first pass of both lipid and aqueous metabolites of the entire brain to see what we could pick up, with the intention of future samples being more targeted based on what we find. So to begin, principal component analysis shows us that our wild type and mutant flies do have distinct metabolomes from one another. They are just empirically different. The first principal component on the x-axis does separate the wild type flies from the mutant flies. Looking more specifically at different metabolites, we can see a couple of different interesting things. First, we see a decrease in several triglycerides containing saturated medium chain fatty acids. All of the triglycerides that we saw increase contained either long chain fatty acids or at least one double bond. We interpret this as being caused by a lack of very long chain fatty acids being broken down, leading to a loss of these medium chain saturated fatty acid breakdown products. We're also able to see an increase in ceramides with saturated C14 and C24. So we know from literature that lipid overload can lead to an increase in ceramide levels, and that increased ceramide levels are linked to neurodegeneration. Our interpretation is that the very long chain fatty acid buildup is leading to an increase in these ceramides. After looking at the metabolome, we also looked at the transcriptome. So we performed RNA sequencing, and to begin looking at a principal component analysis, the samples do cluster based on genotype. Our wild type and mutant flies have different gene expression patterns from one another. Looking more specifically at different specific pathways, gene set enrichment analysis shows us that there is a significant increase in expression of genes from the TOL-IMD pathway. This is the pathway in Drosophila that is responsible for immune signaling. So this increase in gene expression is indicative of inflammation in our flies. We can also see a decrease in various pathways associated with sugar metabolism, including the pentose phosphate pathway, glycolysis, galactose metabolism, and fructose mannose metabolism. So just to summarize the model to this point, ABCD1 mutant flies have shorter lifespans, experience age-related locomotor decline, do develop brain lesions, and they develop larger brain lesions compared to wild type. They have a distinct transcriptome, which includes increased immune signaling and decreased sugar metabolism. And they have a distinct metabolome, which includes fewer triglycerides, which contain saturated medium chain fatty acids. So what can we do with this model? What I've done in this research is test treatment of liraglutazone and its impact on the ALD fly model's phenotype. So most, if not all of you know, liraglutazone is an experimental drug from Minarex, who were kind enough to provide a drug sample at no cost for me to test in this research. And both preclinical and clinical trials have shown a lot of promise, in my opinion, with liraglutazone. And what we did here is treat flies for their entire life with liraglutazone and evaluate their ALD phenotype with the same assays I've just described. So first, we see that liraglutazone treatment significantly increases the average lifespan of mutant 1. For ease of visibility, the Kaplan-Meier curve does not contain mutant 2. We didn't see any significant change there, but the median lifespan of treated mutant 1 was increased by roughly a week, which is a lot in fly terms. Looking at our climbing assay, we also saw a reduction in age-related locomotor decline in mutant 2 as a result of liraglutazone treatment. Untreated mutant 2 flies have mostly lost the ability to climb by this point, whereas treated flies still retain a fair amount of climbing ability. And as a note, this was done at 6 weeks of age instead of 5 weeks of age. At 5 weeks, we saw a trend, but it was not significant, and we decided to push the assay another week. Looking at the metabolome, we were able to see that there are distinct metabolic differences between treated and untreated flies, with our treated wild type and mutant flies clustering more closely with each other than our untreated wild type and mutant flies. This indicates that the wild type and mutant flies which have been treated become more similar to one another due to this treatment. And then the following data in metabolomics and transcriptomics is still in progress. I have a lot to sift through, but this is some preliminary findings. We were able to see a shift in the ratio of reduced glutathione to oxidized glutathione in our treated flies. So glutathione is an antioxidant, and ratios of reduced to oxidized glutathione can be used to measure oxidative stress. Lower ratios indicate more stress. When liraglutazone is given to these flies, we see that they have lower levels of oxidative stress. And I will note here, the wild type does show oxidative stress. I am aware of that. I haven't had opportunity to explicitly confirm what's happening, but I believe it's due to the fly's age. These are 5-week-old flies, and it has been shown in literature that aging flies do experience oxidative stress. That's something that I'll be following up on in future work. Looking at the RNA sequencing data, principal component analysis shows us that there are transcriptomic changes associated specifically with liraglutazone treatment. And you can see that the first principal component separates our samples based on genotype and the second based on treatment status. You can also look at the spread of the data points and see that the treated and untreated mutant clusters are further apart than the wild type treated and untreated clusters, meaning that there's a bigger difference in the meaning that liraglutazone has a greater effect on mutants than on wild type. Looking at some of the pathways affected by this, overrepresentation analysis shows us that liraglutazone is affecting lipid localization processes, which encompass transport, storage, and location maintenance. So just making sure it stays where it's supposed to be. Mutant flies treated with liraglutazone have reduced expression of lipid localization genes. Upcoming lipidomic analysis that I will be completing will help us identify if any particular lipid species is reduced in conjunction with this reduction in lipid localization genes, but right now we can't really say much more than that. Liraglutazone also leads to a reduction of genes involved in immune signaling. If you'll remember earlier in this presentation, we saw that mutants have a higher expression of immune signaling compared to wild type. We can see here that liraglutazone treatment has partially reversed this inflammatory phenotype. So in summary, liraglutazone treatment increases lifespan in mutant 1, slows age-related locomotor decline in mutant 2, reduces oxidative stress as measured by ratio of oxidized and reduced glutathione, is associated with decreased expression of genes for lipid transport, storage, and location maintenance, and is associated with a reduction in immune signaling. Treatment with liraglutazone has partially reversed many of the phenotypes associated with ALD in our model. Hopefully further analysis will give a clearer picture of what the potential benefits of liraglutide treatment are on a molecular level. Finally, for next steps, I will be completing the liraglutide brain histology. I really wanted to show that to you guys today, but it just did not work out. But that's what I'll be doing once I get home. I'll also be cross-referencing the findings from the RNA sequencing and metabolomics data, as well as continuing to analyze these data sets on a deeper level and analyze data as it comes in, because I am still receiving data in. We will also be identifying potential metabolite deficiencies in our metabolomics data in order to see if supplementation of these deficient metabolites back into the diet can be used as a treatment. Finally, we are also looking for a collaborator who works with iPSCs to be testing these dietary supplements as well. So if you or your lab may be interested, my contact information will be on the next slide. So with that, I would like to acknowledge my lab, as well as the Van Andel Institute Genomics Core, Mass Spectrometry Core, and Optical Imaging Core, my thesis advisory committee. I'd like to thank Minarex for providing the liraglutide and agreeing to collaborate with me. And I'd like to thank ALD Connect for funding and for allowing me to be here and talk to all of you today. Thank you.
Thank you, April. I got to say, this photo looks like an alternative rock band, not a science research lab.
Who's to say we're not?
Guys are pretty cool. All right. Any questions for April? All right, we got one over here.
Yeah, why did you use two different mutants? Are they, is it not known which might be more similar to humans?
No, so they're both insertion into the gene to cause a frameshift mutation, and we saw that both of them had a loss of ABCD1 expression. The point of it was to see the effect of ABCD1 mutation in different genetic backgrounds. Generally with fly lines, they're all pretty homogeneous from line to line. So we just wanted to see what multiple different backgrounds would look like in these tests. Is that okay?
Over here, great.
Yes.
Awesome talk, April. I'm going to dovetail on the last question just briefly. In your first PCA plot, it looked like mutant 2 was closer to the wild types, whereas on the functional data, it looks like your mutant 2 was actually quite affected in comparison. Do you have a reason to believe why those two may be slightly different?
I have no idea. That is
Okay.
That is a problem for me to figure out at some point in the future.
I don't mean to imply it was a problem. I think it's fascinating.
Yep. For the PCA plot thing, mutant 2 is more similar to wild type in terms of genetic background, though.
I have a silly question, but does ALD make you blue?
You know, I did not know about that whole campaign, and then you guys mentioned it this morning, and I wasn't sure if I should say anything about it, but you know, maybe it does.